The third second line of defense is a fever. So a fever, as we all know, is a systemic rise in our body temperature. So it's an internal temperature increase. Normally, our internal bodily temperature is a 98.6, or is at 98.6 degrees Fahrenheit, and that's an average. I mean, obviously that can fluctuate from person to person depending on things like age. But generally an average of 98.6. And then we have low grade and high grade fevers. So a low grade would be 100 to 101 degrees Fahrenheit, and then high grade 104 to 106. And then anything after that can often be considered detrimental or potentially even fatal. So how does our body control our temperature? So we have a region in the brain called the hypothalamus, and the hypothalamus is essentially our body's thermostat. So it is going to control the temperature. And when our body is in an infectious state, it wants to increase the temperature. So when we have an infection, our body is going to release chemicals called pyrogens. And pyrogens are going to travel through the blood to the brain, where they will interact with the hypothalamus, and the hypothalamus will increase the temperature. So when the hypothalamus comes in contact or recognizes that pyrogens are present, that's when it knows that it needs to increase the temperature because the individual is under a state of infection. So if pyrogens are sensed, the hypothalamus will go to increase the temperature to 100. If pyrogens are still present, it could go to 101 and continue to rise as long as the pyrogens are present, because that's a sign that the infection is still there. Once the infection subsides, either from immune responses or medication, such as antibiotics, the pyrogens will be lifted, and then the hypothalamus knows that it can start to downgrade the temperature again back to the standard 98.6. So one thing that I think often is done all too often is to immediately bring down a fever. So I think a lot of times when we're sick and we take our temperature and we see that we have a fever, even if it's just a low-grade fever, our gut instinct is to take something like Tylenol and bring our bodily temperature back down to 98.6. And that's not always the best option. We have to consider that our immune system is initiating a fever for a purpose. It is a natural part of our immune system and is going to help us overcome the infection. Now, this is a kind of a rule if you have a low-grade fever and you're an otherwise healthy individual. If it's a high-grade fever, you should take something to bring it down. If you have any type of fever and you are immunocompromised, or a young child or elderly, you should generally take something to bring it down. But if you are a healthy individual and you just have a minor low-grade fever, you should not take anything to bring it down, because it is your body trying to clear an infection. So we need to talk about the benefits of having a fever. So one of the major benefits of having a fever is that now your internal body temperature is greater than the optimum growth temperature for bacteria. Microorganisms generally prefer to live at 98.6 degrees Fahrenheit. So if your bodily temperature is 100 or 101 because you have a low-grade fever, that's too warm for them. So microbial growth is actually going to slow or completely stop. But if you have a low-grade fever and you decide to take Tylenol to bring it down to 98.6, really you're just creating the optimal environment for the microorganisms to grow and make you even more sick. So we want to increase that temperature because it's going to slow or potentially even stop microbial growth. Another thing is high temperature can deactivate microbial enzymes and toxins. So all those toxins that we talked about in a previous lesson, like the hemolysins that destroy red blood cells, the leukocidins, the neurotoxins, the enterotoxins, typically those are going to become, or they're going to be deactivated in situations where there's an increase in temperature. They work their best at 98.6, but at higher temperatures, those toxins are going to be deactivated, and as we know, those toxins can wreak havoc on our body. And while increased temperatures can slow microbial growth, the high temperatures actually boost our immune system. So in a fever state, your immune system actually speeds up. Immune cells are produced faster. Phagocytic cells can work faster as far as phagocytosing bacteria and destroying them with their digestive enzymes. So this kind of combination of having a fever where the microbial growth slows and the immune system speeds up is what's going to clear the infection that much faster. So overall, again, if you are otherwise healthy and you have a low-grade fever, it's perfectly fine to let that fever run its course because that is going to help you get rid of the infection faster. So the last two things we'll talk about are actually just proteins that are produced in our body that can help us get rid of infections. So the first is complement. Complement, there's really a series of 26 different complement proteins. These 26 different complement proteins are found in our blood. And when we don't have an infection, they are just individual proteins kind of free-floating within the blood. When we do get an infection, though, within the blood, that is going to activate complement proteins. And when complement proteins become activated, they come together, all 26 of them come together and form this ring-like structure called a membrane attack complex. This membrane attack complex is very important. It's going to sit on the surface of a bacterial cell and essentially poke holes within the cell membrane of the bacteria. And when the membrane attack complex pokes holes in the cell membrane, that's going to cause the internal contents of the bacteria to leak out, and the bacteria will shrivel up and die eventually. So essentially, the membrane attack complex is stabbing the bacterial cells to death. But again, when complement becomes activated, it just forms this structure called a membrane attack complex, and that is going to create holes in the cell membrane of the bacteria, eventually leading to cell death. Another thing that the membrane attack complex does is called opsonization. And opsonization is just enhanced phagocytosis. So if a bacterial cell is in the blood and it has a membrane attack complex on it, that just alerts the phagocytic cell that much faster to come over and help engulf the bacteria and destroy it. Certainly a phagocytic cell would know if a foreign pathogen was present because it would see that little PAMP marker on it, that pathogen-associated molecular pattern that we talked about earlier. That little PAMP marker kind of acts like a little red flag that lets the phagocytic cell know that that bacteria is foreign and needs to be engulfed and destroyed. But if a bacteria also has a membrane attack complex on its surface that's kind of working to destroy the cell membrane, that is like Vegas flashing lights that alerts the phagocytic cell to come and assist. So at that point, the phagocytic cell would assist by engulfing the bacteria and degrading it. So it's just enhanced opsonization is just enhanced phagocytosis. The last thing we'll talk about is interferon. Interferon is an antiviral chemical that our body can produce. So when some cells in our body get infected with a virus, they can release this chemical called interferon, which essentially is used as a warning mechanism to alert other cells within the vicinity that a virus is on the loose and that they should prepare themselves. So when we talk about interferon, we kind of generally say it interferes with viral multiplication. So unfortunately, the original cell that gets infected with the virus, it does get taken over by the virus. There's nothing that it can kind of do to save itself, but right before it gets completely taken over by the virus, it releases that interferon chemical to alert all the other healthy cells in the area of the virus, that the virus is present. Eventually that original cell will get taken over by the virus, and that host cell will produce thousands and thousands of copies of new virus that will be released into the body. But the good thing is that interferon has already been released to warn the other cells that the virus is on its way. So it's really kind of a warning mechanism. So here we have this cell. This cell did get infected with a virus. Again, right before it would have been taken over by the virus, it would have released the interferon chemical. So at this point, then, this cell gets taken over by the virus, and it's eventually going to release more viruses into the environment. But what that interferon chemical did is it warns these other cells. So it can kind of work in several different ways. So sometimes when our cells see interferon, it alerts them to produce proteins that, if they become infected with a virus, these proteins are antiviral, and so when the virus enters the cell, those proteins can actually degrade the virus. So the cell would initially get infected by the virus, but then the cell would be able to release proteins that would destroy the virus, so the virus wouldn't be able to take over the cell. Sometimes if the virus infects the cell, if the cell was warned through interferon, the cell can actually induce apoptosis and burst. I mean, obviously in that case the cell would die, but it would die before it releases millions of viruses into the environment. So, we would lose that one, but we would save millions of other cells potentially. And then generally just interferon, when it's in the vicinity, it's going to kind of stimulate immune responses that are helpful at negating viral infections, things that we'll talk about in our next lesson, like natural killer cells. But generally, interferon is a chemical that is released in response to a viral infection. So when a cell becomes infected with a virus, it can potentially release interferon before it gets taken over by the virus to warn other cells in the area so that they can prepare themselves and prevent themselves from being taken over by a virus.